Hello, my name is Okweoluwa Adeshola. I'm a sophomore biology major here at York College of Pennsylvania, and I did my research on extracting antibiotics from soil bacteria. In today's world is a phenomenon known as AMR, which is a situation that's happening where bacteria are now becoming resistant to antibiotics that would usually work against them. Most notorious among these are the escape pathogens. Imagine going to a hospital with like a cut and leaving with a staph infection. Pretty bad, hence the need to try and find novel antibiotics that can fight these escape pathogens. It is known that soil bacteria can sometimes have antibiotics, and so the goal here was to try to extract soil bacteria with their antibiotic. In a lab setting, it is dangerous to use these escape pathogens, and so safe relatives or safe strains of these escape pathogens is a more conducive option. With this in mind, we set out with two objectives. We wanted to extract soil bacteria capable of producing antibiotic, and then we wanted to characterize the soil bacteria as well as the antibiotic product itself. Let me take you through my methods here. I'll go into more detail later on. What we basically did was we began by extracting bacteria from soil, and then we did an assay of the isolate we extracted against safe relatives of the escape pathogens. After that, we wanted to characterize the bacteria, so we did a gram staining of our isolate, as well as a PCR amplification of the 16S ribosomal RNA gene. Then we moved further on to do a sequencing of the whole, an extraction of the whole genome of our isolate. Then we wanted to understand what it was, so we blasted it, the sequences against the database. Then after understanding a little bit about our bacteria, we wanted to characterize the antibiotic product by doing disk diffusion against known antibiotics, as well as running it through the anti-smash database, which I'll explain later on. That was our methods. Let me take you through our results here. We successfully isolated a bacterial isolate capable of producing antagonistic activity, and we called it isolate OA19, which I will now refer to as from here on out. Basically what you see here is isolate OA19 producing antibiotic and having antagonistic activity against both gram-positive bacteria as well as gram-negative bacteria, basically the gram-positive safe strains of the escape pathogens as well as the gram-negative safe strains of the escape pathogens. Most notable here is the large zones of inhibition isolated OA19 was able to create against B. subtilis, which is the safe strain of Enterobacter faecium. After realizing what gram-positive or gram-negative bacteria our isolate produces antibiotic against, we now wanted to begin to understand. We wanted to know the gram-stained nature of our bacteria. And as shown here, because it is pink, our isolate OA19 is gram-negative. If it was purple, it would mean isolate OA19 is gram-positive. After knowing our gram-positive or negative nature of isolate OA19, we wanted to begin to know what particular details about our isolate OA19. So we did our PCR amplification of the 16S ribosomal RNA gene. We used a 1500 base pair PCR primer. So it should have shown in the 1500 base pair ladder region, which as you can see here was shown. We then took the results of the sequencing of our 16S ribosomal RNA gene, as well as the sequencing results of our whole genome sequencing, which we did later on, and blasted out across the NCBI database. And our top hits showed that isolate OA19 is part of the Pseudomonas species family, with Pseudomonas soli being our most top hit. So we had characterized a little bit about our bacteria, and we knew a little bit about it. It was then time for us to try to understand a little bit about the antibiotic product. So our disk diffusion was done against isolate OA19. We used antibiotic disc with known antibiotics that have several different known mechanism of actions to place in competition against isolated OA19. Our rationale here was that if isolated OA19 is resistant to whatever is in the antibiotic disc and whatever mechanism of action that antibiotic has, then there's a chance isolated OA19 produces antibiotic with similar mechanism of action. And as you can see here, our results show that isolated OA19 was resistant to antibiotics with cell wall synthesis inhibition, folic acid synthesis inhibition, as well as protein synthesis inhibition through the inhibition of the 15S ribosomal subunit. After characterizing this antibiotic this way, we sort of were wondering if we could figure out what genes are responsible for the antibiotic production. So we sent our entire genome we sent our genome to NCSMASH, which is a database that picks out biosynthetic gene clusters, which are a pathway for producing secondary metabolites. Antibiotics themselves are sometimes known as secondary metabolites. Our top hits from there showed that isolated OA19 had gene clusters that were similar enough to other known gene clusters that are responsible for producing ptolacin with a 70% similarity, pyroverdin with a 11% similarity, and pseudopyronine with 18% similarity. Let me summarize what I've just said for you. We successfully were able to isolate soil bacteria capable of producing antibiotic from a local dog park here in New York, Pennsylvania. Isolated OA19 was able to show antagonistic activity against a broad spectrum of the escape pathogen safe relatives. Isolated OA19 itself is gram-negative, rod-shaped, and its results of the whole genome blast show that it's part of the Pseudomonas species family. Disc diffusion results show that isolated OA19 may have multiple similar mechanisms of actions to those that it was resistant to. But then when you go to our anti-SMASH data here, ptolacin as well as pseudopyronine are secondary metabolites that affect cell wall membranes, which isn't one of the mechanism of actions that we sort of compared it to. But pyroverdin is sort of interesting. It is a sort of Trojan horse molecule that is used to sneak in antibiotics to bacteria that would otherwise be, ineffective. So my rationale here, my thinking here was probably that pyroverdin, being a Trojan horse molecule, probably has to have some sort of protection against certain antibiotics and things like that. But yeah, in the future, I really hope to work more with pyroverdin. Like I said, it sounds really interesting to me. And I would want to do a CRISPR-Cas9 genome editing on that section of the genome with pyroverdin gene cluster to sort of try to understand more of the mechanism of action of pyroverdin. And also I would like to eventually begin to put together the molecule responsible for antagonistic activity itself through IR spectroscopy, so begin to characterize the organic chemistry aspect of it as well. I'd like to thank God for wisdom, knowledge, and then my law partners, Jacob and Taylor, for their support through this, as well as York College of Pennsylvania for the research grants we were granted. And finally, I would really like to thank SACNAS for the opportunity to present my research.